Hello, my name is Cheryl Quate, and I'm presenting my research. Bioinformatic analysis predicts novel tissue-specific enrichment of nuclear pore and gene regulators. So what is my project actually about? Well, in all eukaryotic cells that have nucleuses, surrounding that nucleus is the nuclear envelope, and lining that nuclear envelope is a structure known as the nuclear pore, which is at the heart of my project. I'll be referring to it as the nuclear pore complex, or the NPC. The nuclear pore complex, we know of it as a transporter, and it allows materials to pass between the inside of the nucleus to the cytoplasm. And it's constructed of about roughly 30 proteins called nucleoporins, or NUPs, which is the abbreviation. It's also made up of 6 different subcomplexes, which are seen in this figure here to the right. Those subcomplexes are the cytoplasmic subcomplex, the inner ring, the central channel, the Y subcomplex, the transmembrane, and the nuclear basket, and the constituent proteins for each of these subcomplexes is also listed in this figure. Research has shown that the nuclear pore doesn't only work with transport, it has very important roles in things like DNA repair, cell differentiation, telomere maintenance, and even things important like gene expression. So for a structure that's found so ubiquitously throughout almost all of our cells, researchers have begun to theorize that potentially having a well-regulated nuclear pore is important to having well-regulated life. So a contemporary wave of research has started looking at the nuclear pore complex in the context of disease research. What's interesting, though, is as they've conducted this research, they see that certain mutations in nuclear pore proteins lead to tissue-specific disease. This can be seen in the example of a mutation in the nuclear pore NUP155, which manifested in the clinic as atrial fibrillation. So NUP155 is a protein that's found in basically all the cells and all over our body. However, it manifests as atrial fibrillation, which is very centralized to just the heart. This very tissue-specific response from nuclear pore knockouts and mutations points to the idea that maybe the nuclear pore complex is being regulated or is just composed differently, depending on what tissue it's in. Which leads me to the goals of my project, which was to use gene expression data to verify whether or not the nuclear pore complex is being regulated and coordinated differently at the gene level, depending on what tissue it is, and then to take it a step further and then use bioinformatic processes to predict what are the actual potential regulators that are causing this. Which leads me to my main hypothesis. Individual factors coordinate nuclear pore and gene expression in a tissue-specific manner. I'd also like to point out that this is a gene expression project. That is not to say that no other things could possibly be controlling the composition of the nuclear pore complex, such as protein-protein interactions, but this project is limited to mainly just gene expression. So how did I start my project? I needed a lot of gene expression research, and because I conducted this entire project at home, I needed to get that from the internet. And I found this amazing website called the GTEx database, which is based out of Australia, which houses over 17,000 tissue samples from over 900 donors, generally from organ and tissue procurement organizations. And what GTEx does is essentially it will get a profile for the level of expression of a gene in a certain tissue. And it does this in 54 different tissues, and I used 52 for the research that I conducted. I took that data and I generated it into the form of a heat map, which can be seen over here in this second box. The names of the tissues are listed here on the right, and at the bottom are the names of the nucleoporin genes. The shading of the box indicates how much expression is happening. For example, darker shaded boxes means that there is more expression, which is calculating median transcripts per million, and the lighter shaded boxes means that there is less expression, which is less median transcripts per million. Using those heat maps, I looked for different trends, even trends that were happening between different nucleoporins, and I noticed that some of the nucleoporins had very similar patterns of expression, which indicates possible co-regulation. To visualize this, I used this cool tool called a circus plot, which can be seen, which is this circle right here. This circle right here is actually called an ideogram, and on the periphery are the numbers of the chromosomes, 1 through 22, X and Y, and on the inside are the names of the nuclear pore proteins and their location down to the megabase on which chromosome they are. The links in the center represent possible co-regulation between nucleoporins and possible long-range interaction across the genome. Beyond that, to the part where I wanted to predict the actual regulators of the nuclear pore complex, I needed a list of general regulators. So I took myself to the GeneCards website, which houses a tool called GeneHancer, which will give you roughly 200 to 300 potential transcription factors for each gene that you choose. And I did this for all 30 of my nuclear pore proteins. It does this based on a couple of different metrics. Some of the important ones are the gene association score, which essentially GeneHancer will take a nearby transcription factor binding site close to your target gene and record all of the transcription factors that are associated with that binding site. And then it will multiply it by a confidence score, which is essentially how many times have we seen this in different sources, which gives you a main total score, which is what I use to select my transcription factors. And this gave me a very long list of transcription factors, and I wanted to prioritize certain transcription factors that were the most likely ones. So I conducted a Venn diagram analysis to find overlap between transcription factors within the same subcomplex and the overall nuclear pore complex, the idea being if there are transcription factors that I'm consistently seeing and that they are conserved, they're more likely to actually be the transcription factors. Using this list of prioritized transcription factors, I wanted to see if these transcription factors would appear on lists of genes for very specific tissues. To do this, I went to the Gene Ontology Consortium website, which houses lists of genes for specific tissues that are conserved and enriched throughout multiple species. For example, the heart has roughly 600 very, very important genes. I wanted to see if there were any of my transcription factors would appear on the list of these very, very important heart genes. And I did this for 4 different tissues. I did this for developmental heart tissue, developmental liver tissue, kidney tissue, and brain tissue. So on to my results. My first result being that nucleoporin gene expression differences occur throughout unique tissue. This right here is just a completed figure of the nuclear basket subcomplex showing the heat map and the Circos plot. It's also the nuclear basket region is also located right here, which is from the first figure on my slides, but I'd actually like to draw your attention to the heat map. So on the heat map, the tissues that I use are located on the bottom, and the names of the NUPs are located here. What we can see from this is that different tissues have different levels of expression for each NUP. So, for example, we can see that the shading is a lot different for skeletal muscle tissue than it is in, say, lung tissue. There is differences in how the nucleophorins are being expressed. What's even more interesting is that tissues within the same organ have different levels of expression. We can see that within the brain tissue, which is at the bottom highlighted in yellow, we can see that levels of expression in, say, the hippocampus are different in, say, the hypothalamus, or the medulla of the kidney has different levels of expression than the cortex. This kind of verifies that the nuclear pore complex is different depending on what tissue it's in. My next result was that there is potentially co-regulated nucleoporins and that the interactions that they would have would be long range and trans-chromosomal. So this right here is a figure of the inner ring subcomplex showing the heat map and the Circos plot. So on this heat map, we can see that NUP205 and NUP188 have very, very similar patterns of expression. This indicates that they're possibly co-regulated. However, when we look at the nucleoporins on the Circos plot, we see that NUP188 is located on the 9th chromosome, while NUP205 is located on the 7th chromosome. This means if they are actually co-regulated, then that means they're interacting across the genome, and that's transchromosomal interactions occurring. All of these possible co-regulation links are also listed in this table right here to the right. And this slide right here just shows all of the completed figures for each of the 6 subcomplexes of the nuclear pore complex. On to me trying to predict those regulators. So this table here shows my scoring process on the GeneHancer website to find my transcription factors. This column right here shows the number of transcription factors that I was given by GeneHancer for each of the different nucleoporins. The total score is listed here in the center column, which I used to select these. And I would also like to point out that this column right here shows the GeneHancer identifier, which can be plugged in, and you can find the exact same data that I did using the GeneCards website. Then I did my Venn diagram analysis, which looked for overlap between transcription factors that I saw through different nucleoporins. For example, B shows the central channel. And in the center of this, we can see that there are 139 transcription factors that are seen in NUP62, NUP58, and NUP54. And the specific numbers of transcription factors that were conserved for each of these subcomplexes are listed here. For the Y complex, 91, the transmembrane complex, 117 regulators, and so on. But when I took all 30 of the nucleoporins across subcomplex, I found a total of 36 transcription factors that were conserved to regulate the entire nucleopore complex. And these are essentially true hit transcription factors that are very conserved. And using that list of 36 transcription factors that I found, I crossed those with lists of enriched genes in 4 different tissues, which were heart tissue, liver tissue, kidney tissue, and brain tissue, to find the overlap if there are any enriched genes, regulator genes, for these specific tissues. And those specific genes are listed in this table right here in the center. For the heart, I found 4 different regulators. For the brain, I found 5, and 1 each for the liver and the kidney. And overall, there are 11 of these factors, 10 of which are unique. I'd also like to point out that the regulators for the nucleopore complex in each tissue are different. This means that the nucleopore complex has different regulators depending on what tissue it's in. This kind of harkens back to the idea that the nucleopore complex has very tissue-specific regulation, which has a lot of implications for how we look at nucleopore complex disease research. So when we would do disease research, we want to look at how is the nucleopore complex being differentially regulated depending on what tissue we're looking at. So then on to my conclusions. First, we found that the stoichiometry of the nucleopore complex is to some extent coordinated at the gene level. This is what we found from our GTEx heat maps that showed that there was different expression for the nucleopore proteins depending on what tissue it was in. 2, we found that the nucleoporins are potentially co-regulated transchromosomally. This is the takeaway we saw from our Circos plots, which showed that if co-regulation is occurring, then there is long-range interactions for that to occur. 3, we found 36 essentially true hit transcription factors using my bioinformatic algorithm. I did not go into a lab to find these transcription factors, so these are predictions, and we found 36 of them. And 4th, out of those 36 true hits, we found 10 unique transcription factors predicted to regulate the nucleopore complex in my 4 selected tissues, which are seen in this table to the right, which were heart tissue, brain tissue, liver tissue, and kidney tissue. Finally, I would like to give a huge shout out and a lot of appreciation to my lab. I would like to thank my mentors, Dr. Randolph Faustino and Dr. Claudia Preston. I could not have done my research without them, and I would like to thank them for all the advice and all of the help they've given me throughout this process. Thank you.
Hello, everyone. My name is Rishabh Jain, and I'm from Westview High in Portland, Oregon. Today, I'm presenting my research on an artificial intelligence-based codon optimization tool to improve protein expression towards efficient vaccine manufacturing. Today, I'll introduce you to the codon optimization problem, my objectives, my research methodologies, statistical data analyses, and finally, some discussions and conclusions. So to resolve the COVID-19 pandemic, ramping up the production of vaccines became paramount, and I wanted to see if I could make a difference in this field. Recombinant technology is an emerging field that has quickly placed itself as a keystone of synthetic biology. In this method, researchers can take a custom designed DNA fragment and then use it to create a recombinant DNA plasmid. And when introduced inside of a host cell, such as E. coli, the desired protein is produced by the host. After purification, this output protein can then be used to form functional pharmaceuticals and vaccines that are more cost-effective than traditional techniques. Now, when designing the recombinant DNA plasmid, synonymous codon usage bias arises. There are 64 codons, each of which codes for an amino acid. However, there are 22 amino acids. This results in synonymous codons, where multiple codons may code for the same amino acid. And inside of the ribosome, although the same protein is produced, the synonymous codons are not redundant. Low-frequency codons end up taking longer to be translated due to less tRNA being present in the host for that specific codon. By using some codons over others, like for instance, the UAC codon instead of AUC, there can be massive effects in the recombinant expression, up to 1,000 times. If we design proteins with certain codons over others, translational efficiency improves, decreasing the cost of the end product and improving the overall efficacy. Current solutions are led by biotech companies like IDT. However, these tools have a major drawback. When optimizing the synonymous codons of the specified amino acid, they match codon frequency based on the codon usage bias of the target organism. Or, for example, use lookup tables that replace all synonymous codons with just a single one. This ends up introducing cellular toxicity because rare codons and subsequences are neglected that may have had a significant impact on expression. And further, within the cell itself, tRNA pools become imbalanced and metabolic stress is imposed. I wanted to develop a codon optimization tool that takes an amino acid sequence and optimizes it based on the sequential and contextual information present in the genome of the target organism. My goal was to train on a robust data set of over 10,000 genes. My tool should have less than a 1-hour runtime for optimizing the sequence. And finally, I based my solution on recurrent neural networks, as this algorithm can understand sequential context. My methodology was split up into 4 main parts: curation, encoding, learning, and analysis. In the curation step, I retrieved over 6.8 million genes from the National Center for Biotechnology Information, or NCBI. However, I noticed something. A lot of the genes that were present were redundant and or hypothetical. This is where I ran into my first engineering roadblock. To reduce redundancy, I was using a combination of custom scripts and CD hit. However, 6 gigabytes of proteins were too much to hold in memory. I created more custom scripts in MATLAB and macros to create an iterative batch process. And after repeating these steps iteratively, I arrived upon 42,266 sequences, which were over 4 times more than my goal, and they were non-redundant and clean, ready for the next part of my methodology. So over the last couple of years, I've been learning about natural language processing, or NLP, which is one of the most fascinating cross-sections of linguistics and AI to me. To train the recurrent neural network, my data had to be encoded as a set of time steps. Each time step would represent an amino acid in the sequence. I experimented with 2 main types of encoding: one hot encoding and nonlinear Fisher transform from physicochemical properties of amino acids. It was really interesting to see how these amino acids could be encoded in different ways, as if they were just numbers. Now, this brings me to my favorite part of the research: deep learning. Deep learning is an artificial intelligence technique that offers an abstract way of prediction by using artificial neural networks. First, I split my data into 3 buckets for training, development or validation, and testing, with a 70-10-20 split. My model would then be given amino acid encodings for each sequence. Then its goal was to predict the actual E. coli sequence. Now, by doing this, the model is trained to predict and understand sequential and contextual information present in the E. coli genomes. And this is really important because learning the patterns of E. coli will offer the most optimized gene approach. For development, I used my RTX 2060 with an amazing 1920 CUDA cores. And for my final model revisions, I used a high-performance computing cluster with the NVIDIA Tesla V100 with an amazing 5120 CUDA cores. Now, this is what I like to call Codonify Net V3. It takes an input of an encoded amino acid sequence, And as you can see, I used 4 BiLSTM and dropout blocks, which arrived after hundreds of hours of refinement and engineering. Then the model will output an optimized DNA sequence. I've been learning about AI for the last 5 years, and I've learned that the process of tuning hyperparameters is a combination of engineering and art. This is the real art, the art of hyperparameterization. I had many variables that I experimented with, things like mini batch size, LSTM layers, hidden units, learning rate, and more. I made many engineering trade-offs like trading complexity for memory to overcome roadblocks that I encountered along the way. Now, since this is really the basis of life, it's difficult for even a machine to learn this, but not impossible. After fine-tuning my model and improving it bit by bit through thousands of iterations, my model was able to learn the complexity in the host genome. I use various statistical techniques to quantify the performance of my tool, such as the codon adaptation index, which is a metric commonly used in codon optimization problems. Now, when a reference set of highly expressed genes is used, CAI indicates high expression. And on a broad set of 8,000 genes, my tool Codonify offered an increase in CAI of 0.15. When extrapolated to findings by Dos Reyes et al., Codonify offers an average of 236% more expression than the original sequence. This can produce far more vaccines and drugs in a shorter amount of time. Now, while this may look like Starry Night by Van Gogh, or maybe just some wiggly lines, it is the heart of my solution and exemplifies the most beautiful outcome of this research. As can be seen, the various iterations of Codonify offer a significant improvement in CAI compared to the unoptimized or original sequences. However, even for genes that may be less expressed, Codonify doesn't just overwrite the frequencies. It still maintains some rare codons and subsequences in the gene that affect expression. Now, this figure contains pie charts representing the various codons present for each amino acid. Now, I found this part really interesting. Codonify preserves many rare codons that may be important for expression as opposed to other techniques. For instance, the IDT tool uses AGA primarily for arginine, and 5 reference papers show that this codon is associated with negative expression. But by understanding sequential information and optimizing based on the host genome, the AGA codon is replaced by a variety of other synonymous codons by Codonify in the FALVAC1 gene, which is a malaria vaccine candidate. When compared to the industry standard, I like to use the analogy of computer passwords. Let's say that your password is a random combination of A's and B's. If I know that 50% of your password is A and 50% is B, this doesn't really get us closer to guessing your password. But when we learn the sequential information behind it, like learning the underlying patterns, for instance, AABA, we come a lot closer to learning the true bias. Now, doing a case study on FALVAC1, I also analyzed biological principles like negative CIS and repeat elements, which often harm translation. Codonify was able to reduce the amount of negative repeat elements and negative CIS elements. I also developed a modular user interface to create a solution for a potential user. So in this research, I went through countless revisions and used the advanced software high-level development flow to develop my tool. And I'm very excited because I'm currently in discussion for licensing Codonify with a biotech company. I've also been able to validate my research with them. To further validate Codonify, I surveyed over 300 prominent experts in synthetic biology and got great feedback on my problem statement and got a voice from the customer to understand conditions of use. So in this research, I was able to design a codon optimization tool to understand sequential information using a novel recurrent neural network approach. Codonify offers a significant improvement in CAI, which correlates to an enhanced protein expression, which can make vaccines more cost-effective and allow them to be distributed quicker. I've applied Codonify to a Plasmodium falciparum vaccine, which is the most deadly malaria parasite. And I'm very excited about the results I've received so far, so I am not stopping here. In the future, I plan to add the ability for Codonify to optimize sequences based on other organisms, such as mammals and yeast. Further, I plan to increase confidence in Codonify further by testing my model in the lab. I'd like to thank my mentors and teachers for their support, as well as these experts for their invaluable help. Thank you.
Hi, I'm Sahasra Pokunuri, and my project is a novel Alzheimer's disease therapeutic model attenuating hyperphosphorylated tau and amyloid beta aggregates by characterizing antioxidative, anti-inflammatory, and neuroprotective properties of natural extracts. So Alzheimer's is a neurodegenerative disease that is the sixth leading cause of death and currently affecting 5.8 million people in the United States. The hallmarks of Alzheimer's are two specific proteins, beta amyloid and tau, and they drive the disease's infamous symptoms like memory loss, behavioral issues, and problems thinking. And so these protein-rich plaques and tangles degrade the brain beyond repair. While there is no cure for Alzheimer's disease or a way to stop or slow its progression, there are drug options that may help treat symptoms. However, the neuroprotective effects of the plant extracts in animal and clinical studies involving Alzheimer's disease are well under-researched, and so the long line of drugs for Alzheimer's from Regeneron, Biogen, Roche, Merck & Co., and Eli Lilly did nothing to slow or stop cognitive decline for Alzheimer's patients. And although some extracts were studied in relation to Alzheimer's, there are many extracts that were under-researched, and to the best of my knowledge, a combination of plant extracts was not tried to delay the Alzheimer's. Many research reports indicate that oxidative stress and neuroinflammation are the key pathways implicated in the pathogenesis of Alzheimer's. So patients facing Alzheimer's need the research and solutions now more than ever to treat or delay the progression of this disease. And so since these pathways initiate the abnormal deposition of amyloid beta and the accumulation of neurofibrillary tangles of tau protein, which thereby leads to neuronal death, I hypothesize that natural extracts with strong antioxidative and anti-inflammatory properties may be valuable to control neurodegeneration and thereby delay the progression of Alzheimer's disease. So Drosophila melanogaster, or the common fruit fly, has become a model organism and is believed to have roughly a functional homologue of 75% of human disease-causing genes. And so the biological similarities between humans and Drosophila have been exploited with great success in the field of neurodegenerative disease in general and Alzheimer's disease in particular. And so the principal reason behind this is that the fly's brain contains approximately 200,000 neurons. And just like the vertebrate central nervous system, it's composed of a series of functionally specialized substructures. Also, fly tissues are equivalent to many of those found in mammals, including the heart and kidneys, and a high proportion of genes associated with neurodegenerative diseases in humans are expressed in the equivalent fly tissues. And so due to these functional and structural similarities, any lifespan or neurological improvements in disease fly models that we achieve, it's suggestive that they transfer proportionately to other animal models and to humans as well. And so for this research project, I used the Gal4 UAS system to produce flies overexpressing the human amyloid beta 42 transgene in all the neurons. So the Gal4 UAS system, it's commonly used in Drosophila to drive expression of a gene of interest. And so Gal4 is a transcriptional activator that binds to the UAS enhancer sequences found in DNA. And so it then recruits transcription machinery to that site to induce gene expression. And so as a result, genes encoded downstream of the UAS sequence are only expressed when Gal4 is expressed. And so for this study, I wanted amyloid beta 42 in all the neurons. So I took one transgenic fly line expressing the human amyloid beta 42 transgene carrying an Arctic mutation, and the other is a Gal4 line that expresses Gal4 in all the neurons. So kids from these lines will exhibit all neurodegenerative symptoms like motor skills, memory disorders, and lifespan issues. So for all the crosses, I maintained a male to female ratio of 5:10, and for each extract, I used 3 different concentrations of 1, 2, and 3 milligrams per milliliter of fly food, and every experiment was triplicated. With respect to conditions, temperature is always maintained at 25 degrees Celsius, and flies are always maintained under a 12-hour light and dark cycle. I also usually conducted my experiments at the same time every day from 2 to 6 p.m. And so in this phase 1 study, I took 30 natural extracts from organically grown plant leaves and roots that have extensive antioxidative and anti-inflammatory properties. So the natural extracts are cooked with deionized water and mixed with a cornmeal molasses yeast fly food at concentrations of 1, 2, and 3 milligrams per milliliter. So lifespan of the crosses raised on the 30 extracts is recorded for 2 months, and Kaplan-Meier lifespan plots were used to identify extracts that improve the lifespan the most in first-generation Drosophila. So the first plot shows the survival rate of the negative and positive control groups. The positive control group that has Alzheimer's shows a sharp decline from 20 days due to the disease, but the negative control completes the full lifespan of Drosophila, close to 50 days. Now, if you look at all the other plots, the extracts that have prolonged survival rates of first-generation Drosophila were ranked and then later used for the phase 2 study. So all 30 extracts were ranked based on their importance to extend Drosophila lifespan, and I have chosen 12 of these extracts that had the most significant impact in phase 1. I then asked myself, would the sex of Drosophila make any difference in extending the life of Drosophila? So I performed a chi-square test and saw a square value of 0.325, and this prompts me to accept the null hypothesis that the sex of Drosophila plays no role in extending the lifespan. Now, the top 12 extracts were mixed in varying ratios to make different combinations, and these ratios are determined by the contribution of each extract towards extending the lifespan from its standard mean and 1 or 2 standard deviation points away. And so using these ratios as my proportions, I combined the 12 extracts and made 5 different combinations. I called them ALDC series, which is short for Alzheimer's disease control. And so crosses were again performed, and first-generation flies from the same 2 transgenic lines were raised on these combination extract mediums, and then 3 assays were performed to test neurodegenerative symptoms. So the 3 assays are the lifespan, climbing, and single short-term olfactory assays, and these were performed to test the efficacy of the ALDC combinations on amyloid beta 42 overexpressing flies. So in the lifespan assay, the percent survival in the 5 extra combination groups plus the positive and negative control on regular cornmeal were calculated by applying the Kaplan-Meier method, and the difference was analyzed by using the log rank test. And so as you can see in this graph, the control group that were fed the regular cornmeal showed a sharp percent survival decline after 20 to 30 days, and this is due to the untreated and overwhelming presence of amyloid plaques and tau tangles. The experimental group that's raised on the extra combinations, their survival percentage decline is only significant from days 35 to 55, which kind of indicates a significant improvement in the extract-fed fly's lifespan. Upon further observation, it was concluded that ALDC4 allowed for the longest lifespan. Next, to evaluate the motor skills in the climbing assay, I took a calibrated 9-inch graduated cylinder, placed 20 flies from each diet group, and video recorded the flies climbing for 2 minutes. I then repeated the event for 6 trials, and I analyzed the video to count how many flies climbed past that 7-inch mark. Also, during the experimental process, it was noted that the flies fed the plant extract combinations seemed to be more alert, whereas the positive control group was excessively lethargic and showed minimal energy, especially towards the later days of its lifespan, so around day 30. And so as you can see in these graphs, I recorded decreased motor activity in 15% of 10-day-old flies, 30% of 20-day-old flies, and 60% of 30-day-old flies that were fed regular cornmeal. However, in the fly groups fed the plant extract combinations, only 35% showed a decreased motor activity at 40 to 45 days of age. And so similar to the lifespan assay, ALDC4 showed the least motor activity decline. To evaluate the effects of extra combinations on memory impairment, I built a single olfactory memory assay. One of the key components of the T-maze is a shock chamber, and this is a small cylindrical structure that has alternating copper strips to give the shock. The chamber is connected to a small circuit that uses a capacitor to deliver a shock to the flies. And so 20 flies raised on each mixed plant extract food medium were trained to associate an odor with a 65-volt electric shock. I use the T-maze where the flies are first exposed to valerian root odor, along with the shock for 1.25 seconds and a brief rest time for 3.75 seconds. And so after repeating this cycle for 60 seconds, I move the flies to the second level, where they are exposed to germanium essential oil odor without any shock for 60 seconds. I then move the elevator down to the choice point, where they are exposed to both odors without any electric shock for around 2 minutes. And so the distribution of flies towards each odor was recorded, and performance index for each odor was calculated. So performance index is calculated by subtracting the number of flies that selected each odor in the choice point and dividing that by the total number of flies. For example, a higher performance index would indicate that a greater number of flies selected the correct odor, and this would imply that flies were able to use their associative memory to determine the correct odor. And so looking at the performance index graph of the 30 milligram extra concentration, although the cornmeal-fed amyloid beta 42 overexpressing flies' performance index started falling from day 15, extra combination-fed flies had a gradual decline in performance index, which only became prominent around day 45. The improvement is most prominent in the fly group fed the plant extra combination ALDC4. Similar improvements were also recorded for both the 20 milligram and 10 milligram extra concentrations on all three assays, and this helped me decide that the higher the concentration of the extract, the better the lifespan, climbing, and memory or learning ability. This trend was also noted in the climbing assay, since the flies fed higher concentrations of ALDC4 performed better than the ALDC4 flies fed lower concentrations. So in patients with Alzheimer's disease, it's been reported that the aggregation of amyloid beta 42 proteins occurs during the early stages of neurodegeneration. And so the results of the present study indicate that the wealth of many therapeutic agents like polyphenols, hydroxynamic acids, flavones, and procyanidins found in these extracts have anti-Alzheimer's disease effects in amyloid beta 42 overexpressing flies. The improvements in lifespan and memory abnormalities in amyloid beta 42 overexpressing flies is thought to have been clearly caused by the suppression of amyloid beta 42 aggregation and oxidative stress, and they are protecting the brain against beta amyloid-induced neurotoxicity and neuronal damage. Now, looking at the results of this study, the data clearly supports that plant extract combination-fed flies showed improved motor skills, memory, cognitive, learning abilities, and lifespan. And so these improvements are clearly due to protection against neuronal death by suppressing amyloid beta deposits and reducing the accumulation of neurofibrillary tangles, which thereby delays the progression of Alzheimer's disease. So although the results of this research were very promising, I really wish I had access to transmission electron microscopy to see the plaques and tau reduction in the fly brains that were raised in the extra combination mediums. It also would have been amazing if I could establish that the compounds in these extracts were able to penetrate the blood-brain barrier, and this would trigger many innovative treatment options using natural extracts. Thank you for your time.